So um, let's get started. So today I'm going to be talking about single-cell RNA-seq. We're mostly going to be talking about like sort of the biology and the experimental aspects. We're going to do the sort of first step of the analysis, the sort of bioinformatics part. But that actually ends up not taking too much time to actually get running. It'll take a while for it to actually run. In a second, we're going to start with that and get it running, and then we'll let it run while I talk, and then we'll go back and look at that. Should finish in about an hour, hour and a half, for one sample. And so we'll come back to that later, but I want to get that running. Yeah, so I'm gonna, I have a slide, my next slide, but I'm going to walk through this so you can take a look at this while I am switching to my web browser. There we go. So let me switch. Okay, so everyone should sign into Open On Demand, which is, I will drop it in the chat. Right there. And you've all done this before too, so let me make this a little bit bigger. So of course, when you sign in, you should see this. Anyone not ready for me to keep going? Okay. So I'm going to click up here, which will get me here to the where we can choose which app we want to use. And as usual, we're going to do the CHSI RStudio workshop. And 4 hours should be plenty of time. We're going to change the CPU to 16. So we want to be able to use 16 CPUs at a time. And this is Cell Ranger, which we're going to run is pretty resource intensive. So 16 CPUs, and we want to change the memory to 64. And that should work okay. And then for account, probably most of you, it should default to HIVR25 2024, but if it doesn't, if your account says something different, please be sure and change it. And then for partition, again, it should default to CHSI high, but if it doesn't, then please change that too. So 16 CPUs, 64 gigs of memory, HIVR25 2024, CHSI high, and then for Singularity Image, it should also default to the R25 HIV/AIDS workshop. Anyone not have that? Anyone need me to wait until I wait to do this? Okay, so I'm gonna do launch, and we'll wait for that to open. Okay, and so now I've got it's running, got my connect to our studio server button, so I can click on that. Okay, and I am not in a project right now, so if you're already in the the HIVR25 project, you should be all set. But I'm gonna switch to the project. So I'm clicking on the project button up here, and I am looking for this 2024-2025 HIV workshop. Yours might not have the tilde there. It might just have the name of the workshop. And now it should take a second to open. Okay, so anyone not here? Okay, I'm gonna close this, and I'm gonna close this, and I'm gonna close this. And the next thing we need to do is probably everyone needs to update their repo. So if you want to click on the Git tab and then click on the pull button, and you should see it'll kind of spin its wheels for a minute, and that will download all the updates that we've made in the past week or so. And then you can click on close once close is an option. Okay, so is there anyone not there? Great. Okay, so then the next thing is we need to find the cell ranger script, and that's going to be in here. Sorry, and I'm in the. If you're not the top level, you can click on the R cube there, and then click on Workshop 3 Bioinformatics, and then click on Lectures, and we want the 07_day3 cell ranger demo.rmd. So I'm going to click on that. And that should open up here. And so I'm gonna, I'm just gonna start this, mostly start this running, and I'm mostly going to start this running, then we'll like talk a little bit more details later. I see someone had a computer problem. So I think if you're asking, so this is where things are. If you click on the R cube, that'll get you to the top level of the project. And then you can click on Workshop 3 Bioinformatics, Lectures, and it's this last one. Well, if it's sorted alphabetically, it's 07day3 cell ranger demo.rmt. Okay, so this chunk should look familiar. We're kind of loading a configuration file that has like some predefined paths. This chunk here is just gonna basically tell us where we're gonna find output. We want that later. We're not so worried about it right now. And this is just to double check that like I said, we need to be running with, we requested the container with 16 CPUs and 64 gigs of RAM. So just to be sure that the sort of configuration is all okay. And this is, by the way, so the data set that we're working with has, I think, 6 samples. We're just going to be running one of them, this sample. Because like I said, it's going to take about an hour just to run that one. So just to give you a taste for what it's like to run it, not next week, in 2 weeks, 2 weeks and 3 weeks, you're going to actually be working with the data, doing the downstream analysis. So you'll work with a full data set. We've already run that separately, so the process data is ready. But this is just to give you an idea of like how to run Cell Ranger. And then this is gonna clicking this is gonna actually run Cell Ranger, and you'll see that it's It's just going to churn for a while. You should see like a green bar right here. And actually, if you open the console, yeah, it doesn't really say anything. So I'll talk a little bit about, we'll go into more details later about what this is actually doing. But like I said, I just want to get it started because it's going to be a while before it's done. Anyone not up on that? Any questions about that? Okay, so I am going to switch back to my slides if I can find them. They just are really hiding. I don't know where they went. Ah, they're there. Okay. Okay. And so, oh, yep, run Cell Ranger. Okay. So, let's talk a little bit, well, not just a little bit. We're going to talk for a while about what, about single cell RNA-seq. So last week we talked about bulk RNA-seq. Again, this week we're talking about single cell RNA-seq. So I want to explain to you kind of what the differences are and sort of theoretically like what information you can get from single cell RNA-seq that you can't get from bulk RNA-seq. So I want you to understand like sort of the benefit of doing single cell RNA-seq, and towards the end I'll tell you like situations in which like maybe either don't need those benefits, so it's not worth the additional cost and trouble, or in which there are also situations in which you just don't really have the option of doing single cell RNA-seq, and so you're just stuck with doing bulk RNA-seq. Okay, so this is imagine we're doing bulk RNA-seq, and last week we walked through sort of the theory and the bioinformatics. Next week you're going to do the analysis, but as I talked about last week, one of the most common applications for bulk RNA-seq is to look at gene expression, and particularly differential gene expression. So, for example, you might have a sample in which you have you might have a sample in which you have a control sample and then a treated sample, and you want to compare the expression of a gene between those samples. Actually, I'm realizing I've given this talk several times, and I'm realizing I wish I'd done things a little differently, but that's okay. Or you can look at, say, two different genes. So here, gene A and gene B. And so imagine you have a sample, and you've taken your sample. It's represented up here. It's let's say a suspension, of cells. Like maybe you're taking blood from a patient, and you have millions of cells in there. You you separate the cells from the blood. You extract the RNA from the cells, and now you've done RNA-seq, and now you have this result, and you see that that gene A and gene B have similar expression. And so great, that's what you get for bulk RNA-seq. But what you need to keep in mind is that you're looking at a whole population, and you're sort of experimentally taking the mean of all the cells, the mean expression values of all the cells in that population, right? So while we have individual cells, we have millions of individual cells in there, we're not getting cell level information, we're getting population level information. And so this is roughly, another sort of analogy that I think is helpful is you can think of, you have a bucket of apples, and and we're taking that bucket of apples and we're putting it on a scale, and you can't tell. I see 3 apples here, but it must be a pyramid, so there's probably 4 apples there. And so we're weighing it, and then we're dividing by 4 apples, and we're getting the average weight of the apples in on that scale, right? So we're measuring all the apples together. We have 4 apples, and so we divide the weight that we measure by 4, and so this is our average weight. But these are not like perfect ideal apples that are all identical. This is not, apples are not a manufactured product where you have an engineer saying, like all our, like say M&Ms are equal mass within a tolerance of 0.1% or something like that, right? These are natural products, and so there's lots of variation. So if we, instead of taking a whole bucket of apples and weighing them all at once and taking the mean, we individually weighed those apples, this is what we'd see. We'd see a sort of random distribution around that, this is the mean weight of all the apples. Here's our random distribution of weights around that apples, right? So we have some smaller apples that don't weigh as much, and we have some bigger apples that weigh a lot more, right? And so sometimes the average weight of the apples is perfectly fine measure, but sometimes we actually want to know, like, okay, what actually is the minimum possible weight? What's the maximum possible weight? What's the distribution of weights in our sample? And so sometimes we want this information and or need this information really to get understanding from. Does that sort of difference between population level data versus individual data? Any questions about that? Okay. So, and this is actually an observation that's been around in statistics for a long time. So, like one sort of clear illustration of this is something called Anscombe's quartet, where now a well-known statistician pointed out that you can, for very different distributions of values, you can get the same summary statistics, right? So this is the line of best fit for the points on this plot. And if you look across all these, it's a little hard to tell here, but if you look across these 4 plots, you can see the line of best fit, this regression line, is the same across all of them. So if all you had was the line of best fit for this data, and you didn't look at the individual data points, you'd say like, oh, okay, well, this data is the same as this data, is the same as this data, is the same as this data. But of course, when we look at the distribution of points, you can say, I mean, really clearly that this data is very different from this data, right? All of them are different, but and there's pretty clearly, whatever the sort of underlying, whether it's biology or you're measuring the heights of buildings or whatever, whatever's underlying each of these data sets must be pretty different, right? And so it's really important to understand in general when you're doing summary statistics, but in particular when we're thinking about things like RNA-Seq, that the values underlying that summary value are important in understanding whatever the sort of natural phenomenon is that's generating those individual values. So hopefully I've convinced you to some extent that a summary value, like mean expression of a gene across a population, can we're this is the whole point of a summary, right? Is we're taking a bunch of data points and we want a single value that represents that population. But sometimes there can be, in the case of RNA-seq, there can be important biology that's actually underlying the that summary value, and just having the summary value, we're potentially losing information. And so, of course, that's true for bulk gene expression. So, for example, these summary values of expression of gene A and gene B can, just like the Anscombe's quartet, we can have different biology underlying different biology that can give us this these summary values. So one possibility is that actually, each of these cells, which these red dots here I'm representing with these red dots plotted here. It's possible that each of these cells have values of expression of gene A and gene B that are very close to the mean, right? The mean here is 50, and so the values of expression of gene A and gene B is maybe like 48. The minimum is 48 and maximum is like 52, right? So that's possible. That's what this value for individual cells, and I've calculated this, will give us these mean values, right? But it's possible that we could get these mean values from a very different distribution of values for individual cells, like this. So that this distribution of values for individual cells will also give us these mean values. But this looks fairly different to me in terms of what the biology is than this, right? This tells me that you have some population of cells that's like fairly uniform biologically, and possibly that there's like a very strict control within the cell on the expression level of gene A and gene B and the shared levels, right? This is that the values are like kind of all over the place, and so the same average for the population, but you have some cells that have really high levels of gene A and much lower levels of gene B, and you have some cells that have really high levels of gene B, much lower levels of gene A. And so this is just like Anscombe made this observation about population level statistics versus like individual measures a while back. The fact that you can have differences in cell level expression within a population, cell level of expression of a single gene or multiple genes within a population. This has been known for over 20 years. This is well, you can tell this is a cover of Science magazine from 2002. And of course, when this research was published, it was like pretty surprising. That's why it got on the cover of Science. So this is essentially a version of like this experiment before there was RNA-seq, and certainly before there was single-cell RNA-seq. And so what you're seeing here is bacterial cells. These are clonal cells, so they're all genetically identical. Every cell here that you see is genetically identical to every other one. And what you're seeing is each cell in this population is expressing two different proteins, one called green fluorescent protein that fluoresces green if you expose it to ultraviolet light, and the other called red fluorescent protein that it fluoresces red when you expose it to ultraviolet light. And so again, these are genetically similar. Every single cell here has the same gene, And so what you're seeing is sort of variation, basically a combination of biological noise and potentially to some extent some interaction between cells and the population. Maybe there's some spatial effects going on here, right? So you can see there are some cells here that are pretty bright green. There are some cells here that are pretty bright red, and then there's some cells here that are bright yellow, and then things in between. So bright green means that it's pretty much just expressing the green fluorescent protein, and it doesn't have has very little to no of the red fluorescent protein. The bright red means that it's expressing almost entirely red fluorescent protein, very little green fluorescent protein. Yellow means that it's really yellow means that it's like kind of in between. It's these cells where it's equal amounts of green fluorescent protein, red fluorescent protein, and then like the different shades of orange and like yellow, greenish yellow and stuff like that is like different places along the spectrum. It's like maybe this cell is kind of orangish yellow, and this cell is more like greenish yellow. And so this was like a really simple illustration of the fact that within a population of cells, because up until this point, at least some people, I mean, like certainly when this paper came out, for me, it was like a huge, like mind-blowing experience. Oh, well, actually like the cells in the population are actually not, uniformly doing exactly the same thing, right? That you have diversity of behavior within the population, right? And this is again a very simple behavior, right? How much of green fluorescent protein you're making, how much red fluorescent protein you're making. But when you take this whole population and you measure it at the population level, you get this. You get similar levels of expression of gene A and gene B, or green fluorescent protein and red fluorescent protein. Everyone okay with that? Questions so far? And so again, sometimes you're fine with just looking at the population averages, but sometimes the population averages are hiding some interesting biology. So another way of thinking about this is that you can have, we started off looking at this bucket of, I'm gonna say those are red delicious apples. I don't remember, but this basket of red delicious apples. But you could have a mixture of, say, red delicious apples and Granny Smith apples, right? And so, if we're talking about weight, maybe like all the red delicious apples tend to be heavier than the Granny Smiths. I don't know about red delicious apples and Granny Smiths. I know that they're like, when I go buy apples, sometimes there's some apples that are just uniformly larger than other apples, right? And so we don't necessarily expect something like this when our population is actually a mixed population of two different types of apples. It's at the very least gonna maybe look something like this. And then it's also possible that not only do we have apples in our population, but we have some oranges. I don't know what are those, limes? That's a lime, maybe that's a lemon. That looks like a peach, right? So our population might look like this. And so if we take all these fruit and if we take the whole basket and we weigh them, and we get the average fruit weight, we're probably missing out on some important differences, right? I mean, these limes I expect to probably weigh fairly different than the apples, right? And so same thing with biological data. Our population could consist of two different cell types, right? And so here again we have like almost identical levels, if we take that whole population, that whole, I don't have my test tube here, right? But this whole population of cells from blood, and we just extract the RNA, and we look at the average level of expression of gene A and gene B within that population, they look about the same. But if we can look at the single cell level, and we have a mixture of two different cell types, it could be that cell type 1 is expressing gene A at very high level, and almost zero expression of gene B. And cell type 2 might be expressing gene B at a very high level and almost no expression of gene A. But this situation at the bulk RNA-seq looks like this, right? And so at the bulk RNA-seq level, this is indistinguishable from this, which is indistinguishable from this. So only when we can look at individual cells within a population can we potentially recognize something like this, which again, like the biology going on here, I'm going to argue is fairly different from the biology going on here or here. Is that clear? Questions about that? Okay. And we can, there are all sorts of other ways that we can think of different sort of distributions of gene expression for in different situations that we're going to, just having bulk gene expression data, bulk RNA-seq data is going to be hiding some more interesting, potentially more interesting biology that's going on, right? So you can imagine that we're doing an experiment where we're taking cells. We have some cells that don't get any treatment, some cells that get treatment A, and some cells that get treatment B. And this is what the bulk RNA-seq data looks like. But here we're just looking at gene A expression, just to keep things simple. So, the control has some expression of gene A at the population level. With treatment A, we're essentially doubling the expression of gene A, right? So that's like a pretty significant change. But treatment B, it looks the same, right? We also, treatment B is doubling the expression of gene A. So we might conclude, if we're really just interested in the outcome, the effect of treatment A versus treatment B relative to control, we'd say like, okay, well, it looks like they're pretty similar. But we could, if we have single cell data, we could look at the single cell data and realize, okay, well, in the control, all cells are, expressing a fairly uniform level of gene A. Treatment A, you're seeing every cell like a rising tide is lifting all boats. Every cell is expressing gene A at approximately double the level of the control, the control cells without any treatment. Treatment B is actually, for most of the cells getting treatment B, they're actually expressing gene A at the same level as control. It's just that we get a few cells that have much higher expression. We're talking about 100 times or 10 times the expression level of the control, but the most of the cells that are treated with treatment B. And so maybe this is some stochastic effect. Just some cells get affected by treatment B. Maybe this is a subpopulation of cells that's a different, these are limes and all of these are apples. And so, limes respond to treatment B, whereas apples don't respond at all to treatment B. But, treatment A, limes and apples all respond roughly the same. So there are lots of possible explanations, but without the single cell data, you that way. So without the single cell data, look at this and you say like, oh, okay, well, treatment A and treatment B are pretty similar, right? Questions about that? Is this all pretty clear, hopefully? So like basically, the goal of like the last like 30 minutes or so is just to give you an intuition for what the differences that you get between being able to look at individual cells and make measurements of individual cells versus having to look at the whole population. And essentially by taking a whole population of cells and grinding it up and looking at the RNA, you're essentially doing an experimental average, right? You're not mathematically averaging the numbers. You're using the ensemble RNA from the population to do the averaging for you. No questions? Everyone doing okay? Okay. So let's talk a little bit about how we do this, because last week we talked about how do you go from a sample to RNA to sequence data, right? And you can imagine that to go from a sample to like RNA to sequence data, separate sequence data for each cell is going to be a little bit more complicated. And in fact, it is. It's not like a lot more complicated ultimately. It just, it takes a little, it's a little bit more complicated. So the way I like to think about this is that the basic principle is that basically you need to, we talked last week about having index barcodes per sample so we can multiplex our samples. So for single cell RNA-seq, we potentially are going to do the same thing. We're potentially going to have index barcodes for samples, but on top of that, we're going to have a cell barcode so that we can like label in each individual cell with a unique label. And then all the RNA coming from that cell is going to have that barcode on it. So that at the analysis phase, so we're going to sequence all that RNA, and in the process, we're going to be sequencing the barcodes. And then in the analysis phase, we can go back and say like, okay, well, here's the barcode for cell number 535. So like this RNA molecule came from cell 535. This RNA molecule came from cell 9,252. So I'm going to put that in the cell 9,252 bin, and you just go through all the data that you have, and just like we talked a little, we didn't go into detail, but we talked a little bit about how you would separate demultiplex samples, RNA reads to individual samples. We need to do that same thing at the cell level. So let's talk about how that works. Again, this should look familiar to you from 2 weeks ago, 3 weeks ago, I can't remember. This is the structure of one fragment that's getting sequenced. So the black here is the sequence of interest. That's the biological sequence of the RNA that got reverse transcribed into DNA, and then got the adapters on it. So this is the sort of regular adapters. Here's the index barcode that allows us to index separate samples. And so again, I'll show you in a second, but we're gonna have cell level barcodes. So this is what the barcodes look like practically. And again, we talked about how you're gonna, before you even do your library prep, for each sample, you're gonna assign it to an index barcode. And then when you actually are preparing libraries from the samples, as part of the library preparation process, you're going to put, based on this table, you're going to say, okay, sample A is going to get barcode, the adapter with barcode 1 on it. Sample B is going to get the adapter with barcode 2 incorporated in it. Sample C is going to get the adapter with barcode 3 in it. So that at the back end, we can then, look for this barcode and know that that came from control sample 1. This is all ringing a bell. Everyone okay with this? Okay, I'll take that as yes. Okay, so that's the sort of bulk high throughput sequencing. For single cell high throughput sequencing, we basically need more barcodes because we need to, instead of talking about like 4 samples or I think the slide here before I had what's that 8, 9 samples. We're talking about like thousands, tens of thousands, hundreds of thousands of barcodes so that you can barcode each cell. And we essentially need more test tubes, right? Because we need to be able to put the barcode, put a cell in a test tube, put a barcode in that test tube, and get this sort of per cell level barcoding. Right? Does that make sense? That what we need to be able to do here? Okay. So the more test tubes is I feel like it's actually a little bit easier than you might think if you didn't without knowing how you do this. So the more test tubes usually so there are a couple different ways of doing this. Sometimes people will do like cell sorting, fluorescence activated cell sorting, and sort individual cells into wells of a 96 well plate. And that was definitely like in the early days of single cell sequencing, that's how people were doing it. Another approach, which is definitely more common, and this is what's used on the commercial platforms that I know of, is using microfluidics to essentially make these kind of virtual test tubes. So the way this microfluidics device works is that you have inputs coming from the left here, inputs coming from the bottom here, and inputs coming here from the top and bottom. And so what's being flowed in from the left is, well, actually, maybe I'll start here. So what's being flowed in from the bottom up is a suspension of cells. And then what's being flowed in from the left is some buffer and these magic beads that I'm going to talk about in a minute. And so these two meet at the intersection there, this intersection. And so then when they meet, you're gonna, basically the cells are gonna, be mixed into this buffer with a bead. And then when they get to this point, there's like mineral oil being flowed in from both sides. And so the mineral oil will kind of pinch off these droplets. And so that here where you see white, that is mineral oil. And the blue here is a water-based solution, a buffer, right? And sometimes you're going to get cells that are flowing through here, and they're just going to, they're not going to end up in a droplet. That's what these cells are. They started from the bottom, they flow through, and they didn't get in a droplet. But sometimes the cell will get incorporated in a droplet, and then sometimes those droplets will have a bead in them, right? And so that's what this is, right? This is a droplet with a cell and a bead. You can get droplets with a cell and no bead. You can get droplets with a bead and no cell. But really what you're aiming for is a droplet with one cell and one bead, right? And so these end up being test tubes, essentially. These are the virtual test tubes, these droplets with a cell and a bead. Everyone okay with that? Questions about that? Okay. And I should say, I forgot to mention at the beginning, but I'm gonna plan on taking a break around 2, so like 9 minutes. So this is the device for from 10x Genomics that's used for this is like the disposable that's used in the machine for making preparing these droplets. And so one of these basically you can put um, 8 samples there. So each column is a different sample. I think that either the first column or the last column is like for a control. And so you would lay, I've never run one of these machines, so I don't know exactly which is which. One of the wells is going to be the oil. One of the wells is going to be the like, buffer and beads, and one of the wells is going to be the cells. And then, this goes on a machine and it runs. And then at the end, you're going to have your suspension of droplets in oil here at the end that you can like pipette off. Okay, so we talked about the more test tubes. Let's talk about the more barcodes. So I told you that like these are magic beads, and actually, so this is like zooming in on a bead. I need to tell you a little bit more about the beads in a second, but the bead, looking at just one bead, the beads are coated with oligonucleotides, short pieces of DNA that help this magic happen. So what's labeled as R1 here is the this part here. This part of the adapter that's got the sort of standard bit. If you remember from when we talked about high throughput sequencing a couple of weeks ago, this part is going to hybridize to the surface of the flow cell and also serve as a priming site for the sort of initial cycles of bridge amplification. This is the priming site for the sequencing primers, but this whole thing is what's this whole green and red bit is what's being shown here as R1, right? So that's just the regular adapter. And then we have these special things that are for the specific to doing single cell RNA-seq. And actually, I should say, sorry, that R1 here can potentially have like a sample barcode in it too. So the red and the green are what's special to single cell. So the green is what's called the 10x barcode. And that's what I mentioned before. We have the cell level barcode, right? So each cell is going to get this droplet is going to have one cell level barcode in it that's on this bead. And then also we're going to have what's called a UMI. UMIs aren't unique to single cell RNA-seq. UMI is short for a unique molecular identifier. So there are lots of high throughput sequencing methods where you want to have a UMI. And basically this is a like per molecule level barcode. And so what that lets you do is it's possible that you might because many high throughput sequencing library preps involve PCR amplification. So it's possible that you'll sequence the product of one molecule, one RNA. You might sequence it more than once. The UMI, every RNA molecule is going to get a unique UMI, or actually a unique combination. Every molecule within a cell is going to get, every RNA molecule within a cell is going to get a unique UMI. And so if you've sequenced an RNA molecule multiple times, because it got PCR amplified, all those RNA molecules will have the same UMI. And so you can you don't want to actually count that. Let's say you sequence an RNA molecule from a given cell 7 times. You don't actually want to count it as 7 because that came from one RNA molecule. And your goal is to count the total number of RNA molecules in the original cell. So you're going to collapse those down. You're going to put them in one bin that say like, oh, okay, well, these 7 reads all came from the same RNA molecule. So I'm just going to count that as one read from that gene. And then the switch oligo, I'm not going to get into too much, but basically it's just something that's allowing this part to the rest of it to um, uh, it's essentially like a primer for doing the initial amplification of the initial reverse transcription of the RNA molecule. So this again, the 10x barcode, the bead is going to be coated with, you can see these little hairs here. Each of these is an oligonucleotide piece of DNA. Every single bead has the same R1 on it. Every single bead, all of these individual oligonucleotides on a given bead have exactly the same 10x barcode. And then every single of these little hairs here, these oligonucleotides, has a unique UMI. And then they're all going to have the same switch oligo on them. So when we make a droplet that's got a bead in it, all those oligonucleotides are going to be used for the reverse transcription of the RNA. And so all the RNA within this droplet is going to get the same 10x barcode. And so that's labeling it as being from this cell. There's nothing special about this cell itself, but we want to be able to trace all the RNA molecules back to an individual cell. So that we can then say like, okay, well, this is the gene expression of this cell. And then every single RNA molecule in this cell is going to get a different UMI, right? So within that test tube, within that droplet, all the RNA molecules are going to get the same 10x barcode, but they're all going to get different UMIs. So we can actually trace back not just at the sequencing data, not just to a single cell, but actually to an individual molecule. Does that make sense to everyone? If you're wondering, how do you actually get every oligo on the same bead to be the same 10x barcode? It's actually a cute trick. I'm not going to go into it right now, but if we have time at the end and people are interested, I can explain that to you. It is surprisingly simpler than you would think. Okay, so just to give you a feel for like what these look like, so the standard for 10x, and again, I think I said this before, but 10x is maybe I didn't. So 10x Genomics is like the leader in single cell RNA-seq. There are other competing companies, and there are also protocols. Like you don't actually need to buy a machine from 10x or to buy their reagents. You can, there are protocols for doing single cell RNA-seq. You can just buy reagents and build your own microfluidics device. I think they're probably unpatented ways to do it. But 10x is the most common, the most popular company for doing single cell RNA-seq. This is not an endorsement of 10x in any way. It's just to say that it's the most popular. And so the most likely the method that you're most likely to encounter. And so for 10x, these 10x barcodes, which are cell barcodes, are 16 base pairs long. So theoretically, with 16 mers of DNA, you could represent 4 to the 16th different barcodes, which is about 4 billion. And then the UMIs are 10 base pairs long. And so in theory, you can, and those are, again, per RNA molecule. So that's 4 to the 10th, or about 1 million different UMIs. And you might say to yourself, well, that's actually not like, why do you have so much less complexity for the UMIs? But really, the RNA molecules, you got to remember that they're getting a UMI and a 10x barcode. So you only need to be able to distinguish RNA molecules within a given cell from other RNA molecules within a given cell. Between cells, you're distinguishing them by the 10x barcode, right? Or another way to think of it is like this whole thing is essentially UMI. And so you're actually getting 26 bases of to distinguish each RNA molecule in the whole population of cells. So now that we've talked about, actually, maybe I'll just ask any questions. Oh wait, there was a question in chat from the break. UMI distinguishes individual RNAs from the same cell, and the barcode enables identification of which cells RNAs are from. Yes, yeah, exactly. Yeah, so UMIs are actually barcodes. I should explain that. UMIs are well, actually, both of these are barcodes, both the 10x barcode and the UMI are actually random barcodes. So they're randomly generated, but they're both a type of barcode. And this is, you can think, just like we were talking before about index barcodes are like a per sample barcode. The 10x barcode is a per cell barcode, and then the UMI is a per RNA molecule barcode. Hopefully that clarifies that. If not, I can try to explain it in a slightly different way. Okay, good. So any other questions that kind of percolated in people's minds while we were taking a break? Set a timer to remember to take a break. I think I don't want to say this because I'm gonna jinx it, but I think probably we won't. We'll end before 4, but I think we might go for at least another 45 minutes. Okay. No other questions? And I will keep talking. Okay, so I gave this sort of theoretical background. We talked about like what sort of information you can get from a basic gene expression, single cell RNA-seq, that beyond what you get from bulk. And we kind of talked about how you actually are able to, trace RNA molecules back to a single cell. So I just want to talk a little bit about different applications for single cell RNA-seq. What I've been alluding to so far, and actually I should say not just single cell RNA-seq, any sort of single cell sequencing based method. So what we've been talking about so far is RNA-seq, single cell RNA-seq, and mostly I've sort of been alluding to differential gene expression, but it's pretty common to use single cell RNA-seq both for sort of differential gene expression, but also to do what I'm going to call cell typing. So basically to we saw this example before where we have like sort of clearly 2 different types of cells based on their expression. And so you can use the gene expression of each cell to sort of put it in a bin, to like cluster it with other cells with similar expression. And so you can not just get the expression, but you can essentially get like cell typing. You need to take the clusters and then kind of look up in a database like, okay, what cells are do I know are expressing this kind of collection of genes. But usually you're doing the cell typing based on the gene expression data. And then you can look at differential expression, usually of those clusters. You might look between two clusters and say what genes are differentially expressed between them. But you also might look at like usually you cluster all the cells together, and you might look and say, okay, I've got control and treatment A. I want to look at all the cells in this cluster and say, like, what's the difference between control and treatment A in terms of the gene expression? So you can do differential gene expression at the single cell level, or really at the cluster level. One can also do attack-seq. Attack-seq was developed as a bulk method, but it can be done at the single cell level. So you can do single cell attack-seq. Attack-seq lets you identify regions of the genome that have open versus closed chromatin. So like kind of looking at the that helps identify regions of the genome that are maybe not necessarily actively being genes aren't necessarily actively being expressed, but they're primed to be expressed. You can do site-seq, which is combines doing single cell RNA-seq with measuring cell surface proteins. So this is gets you information. The cell surface protein component gives you information similar to like what you get from flow cytometry, but on top of that, you can, for each of the cells, you can measure not just what proteins are on its surface, which is what you get from flow cytometry, but also like the RNA-seq, the gene expression of all the genes within that cell. And 10x has a version. site-seq is like a method that was developed by like a academic research lab, but 10x has a version of site-seq. You can also do BCR, single cell BCR and TCR sequencing. So B cell receptor, basically the antibody that's expressed on the surface of a B cell. You can sequence the antibodies for the B cells in the population. And you do the same thing with TCR sequencing. So like sequence the T cell receptor that's on the surface of T cells. You can also do, so this is actually looking at the sequence of the receptor. There's also a method to look at the specificity. So you can basically probe T cells and ask, like, is the T cell receptor binding to recognizing this specific epitope? And you can also do spatial transcriptomics, where, what we've talked about so far, that, sort of the assumption of single cell RNA-seq is that you have this population of cells that's in a suspension, but you can actually look in essentially on a microscope slide and look at roughly single cell gene expression on a microscope slide for different cells. So you can actually look at the spatial organization of the cells for like an intact tissue. And this is just like a little bit of the kind of a cartoon of 10x, the way they do both this, the site-seq and this TCR specificity is using this, what I want to say, product called feature barcodes, where in addition to sequencing the RNA molecules with their barcodes, you pre-treat cells with some sort of, before you form them into the droplets, you pre-treat the cells with a tagged protein. So in the case of the cell surface, the site-seq, essentially, for site-seq, you would have antibodies that have a oligonucleotide attached to them. That's essentially a barcode for the antibody. So here you can see like we have a yellow antibody that's specific for a yellow surface protein. We have a green antibody that's specific for a green surface protein. And we have a blue antibody that's specific for a blue surface protein. And so all the blue antibodies in this experiment would have one feature barcode, like all the same feature barcode that would identify that type of, let's say this is an antibody that's specific for, I don't know, CD4, right, on a T cell. So the CD4 antibodies are going to have one sequence of feature barcode. Let's say this is a CD8 antibody, so that's going to have a different feature barcode. And so when these get formed into droplets, then the sequencing is set up such that not only do you sequence the RNA, but you actually sequence these antibody-specific barcodes so that you can say, okay, well, here are the different genes that are being expressed by the cell. And also, I'm seeing like a certain level of CD4 antibody oligonucleotide. And so, you know, that tells me that this cell is expressing CD4 on its surface. And I'm also seeing like, you know, CD8 antibody barcode. So this, I know the cell is also expressing CD8 on its surface. And so the same, and this is, you know, how CITE-seq works in general. And then this is looking at the like TCR specificity. You have these sort of like, they're kind of strung together. These are MHC molecules with a specific peptide stuck in them. And you could have collections of these so that you can then probe and see what, you know, you put in a collection of MHC molecules with the oligonucleotide, or sorry, the peptide stuck in them, and you dump them into your sample and let them, you know, bind to interact with T cell receptors on T cells that are in your sample. And then again, like these different MHC conglomerates of different MHC molecules with the peptides in them are going to be, each peptide is going to get a different feature barcode. And I will just point out, so both of these figures were taken from 10x. So like, I know that this is actually not how antibodies work, in case anyone was bothered by that. That's a 10x problem, not not me, but I was too lazy to make a new figure when when they made one for us. Any questions about that? About how like the feature barcodes work to allow you to be able to detect not just like what genes are being expressed, but also these sort of secondary labels. Okay, keep going. I just noticed in my other window that it looks like cell ranger finished, so and we're ready to look at that. It's done. Okay, so I just spent, you know, the better part, more than an hour telling you about like how single cell RNA-seq, you know, gives you this extra information that's really useful and can tell you something about biology that you can't really tell from, or you might miss out on if you're doing bulk RNA-seq. So why wouldn't you do single cell RNA-seq? Well, so the number one reason is that it's expensive. It tends to be a lot more expensive than bulk RNA-seq per sample. I have not priced this recently, and the price has come down somewhat. But the last time I priced it for doing it at the core at Duke, I want to say that kind of, you know, it depends on exactly what assay you're doing and like how many samples you have, things like that. So like kind of the minimum for the cheapest way to do it last time I checked was over $1,000 per sample for doing single cell RNA-seq, whereas you could easily do bulk RNA-seq for I'm trying to think of the Duke core. I think you could do it for under $100 per sample. Again, depending on like, how many samples it is and like what you're looking for. So it is much more expensive per sample to do single cell than bulk. Um, it is very noisy in a few different ways. Number one, because you're looking at individual cells, you're not necessarily capturing all the RNA from, or you're definitely not capturing all the RNA from each cell. So you end up getting what's called dropout, where there might be, let's say, a CD4 T cell. I just mentioned that before. So CD4 T cell, CD4 positive T cell, you expect it to be expressing the gene for CD4. It turns out that actually, if you look at a population of CD4 positive T cells, in many of the cells you don't see, you do single cell RNA-seq, you're not going to see CD4 for a few different reasons. One of them is dropout, just because you might just you're essentially randomly sampling the RNA within that cell, and so sometimes you just won't capture any RNA molecules for a given gene that you expect to be expressed. So that's what's called technical dropout. There's also what's called biological dropout, which is that just because, within a given sort of bulk population of a specific cell type, just because you expect to be seeing a specific gene expressed, doesn't mean that at the snapshot in time where you extracted the RNA, that gene was being expressed. So going back to the CD4 example, so CD4 receptors are fairly stable on the surface of CD4 positive T cells. And so, if you look at a population of 1 million CD4 positive T cells, some subset of them aren't going to be, at the time that you collect your sample, some subset of those cells aren't going to be expressing the CD4 gene, just because they've got enough CD4 protein on their cell surface, and there's a feedback mechanism for regulating the expression of the gene, and they just don't need to be making any more CD4 protein at that moment in time, so they're not expressing it. So that's what's called biological dropout. And those two things combined make it very noisy. Whereas for bulk, because you have a population of 1 million, 10 million cells, you're experimentally averaging over all those cells, and so you're averaging out the noise. And so bulk RNA-seq tends to be much less noisy. So another reason why you might not want to do single cell RNA-seq is that, or another kind of disadvantage for single cell RNA-seq is you're working with small populations of cells. So for 10x, usually in a sample you can get about 10,000 cells in a sample. So that, 10,000 cells, it sounds like a lot of cells, but if you have a complex sample that maybe the cells, some of the cells that you're really interested in are, say, 1% of the population. And, let's say you're taking a blood sample, and in like a normal human being, you're interested in a cell type that's 1% of the population. So if you get 10,000 cells out of that sample, 1% of 10,000 cells, that's 100 cells, right? So that's not that much. And like, of course, there are cells that are even less common than 1%, right? There are cells that end up being like 0.1%, right? So you might not, if you're really interested in just that one cell type, you're either going to have to like sequence a bunch of samples to get enough representation, or maybe you want to do, you could in theory do some sort of enrichment before you do the single cell RNA-seq, or you could do some sort of enrichment and do bulk RNA-seq just on those cells. The sample requirements are pretty tricky for single cell RNA-seq. I've been involved in a few different single cell RNA-seq projects, and the sample requirements have always been a problem. For the sort of most common, again, this is 10x genomics. They recommend that you, or I'm actually trying to remember where the recommendation comes from. I think it comes from 10x itself, and um, also filtering this through the core that we normally work with at Duke. They recommend that your samples be have 80% cell viability. For RNA-seq, you just grind up all the cells usually, and um, you get your cells, you cut out, let's say you're using a blood sample, so you're gonna usually like centrifuge your cells. Um, if you want to like get rid of the red blood cells, there's an additional step to doing that. But then you extract your RNA and you're done. Because for single cell RNA-seq, you need the cells to be intact for this to work. If the cells have lysed, then they're the RNA that was in the cell is just free floating in the suspension. So that's not such a big problem for bulk RNA-seq, but of course for single cell RNA-seq, it means that if the cell lysed, the RNA is going to be mixed in all these droplets. So that's not good. Oops, going too far. So you really need like kind of happy cells, and it can be tricky to usually for single cell RNA-seq, we want to harvest your sample and you want to get it on the 10x machine within a few hours, because you don't want the cells to start doing responding like lysing or just responding to the fact that you've collected the cells and put them on ice. And so it can be really challenging to get good samples for single cell RNA-seq. It can take weeks to months of optimization to get a good sample for single cell RNA-seq. And another negative is if you're really interested in splicing, usually I sort of have touched on this, but often for single cell RNA-seq, you're only getting a small bit of the RNA, like 70 base pairs, 100 base pairs of the RNA. And so for the most part, you're not going to capture splice sites, and you're not going to be able to like get splicing information. These are not the only reasons, only negatives of single cell RNA-seq. There are others. And actually, sorry, I should qualify this because there are now techniques that are even more expensive than sort of regular single cell RNA-seq. But there are now methods that will allow you to get single cell, do single cell RNA-seq and actually get splicing information. I'm not going to go into that today. Questions about this before I keep going? Okay. So I want to think through a little bit, pretty high level detail about the sort of bioinformatics aspects for single cell RNA-seq relative to what we talked about last week, how you do bioinformatic analysis for bulk RNA-seq. So if you remember from last week, what we talked about is that the kind of big picture view in a nutshell of the bioinformatic analysis is that you start off with raw sequence data, raw sequencing reads from your bulk RNA-seq, and your goal is to build this table where the columns are samples, the rows are genes, and then the individual elements of this table or matrix is the number of our RNA-seq reads that you saw for each gene in each sample. So like here, sample 1, we didn't see any reads from gene 1. We saw 5 reads from gene 2. Sample 2, we saw 9 reads from gene 1. We didn't see any reads from gene 2. Everyone, this hopefully rings a bell. Anyone need me to like go over that again? Okay. So single cell RNA-seq is really pretty similar. It's just that we have to do, as I touched on before, we have to do like sort of a few levels of demultiplexing, right? Instead of just demultiplexing the reads to the sample level, we're demultiplexing reads to the cell level. So then our columns here are really cells, and then those cells are subsets of samples. Rows are the same, rows are just genes, right? And so we're looking at cell 1, cell 2 within sample 1, so cell 1, cell 2, cell 3, cell 4, cell 5, cell up to cell n for sample 1, cell 1, cell 2, cell 3 for sample 2, cell 1, cell 2, cell 3 for sample n, right? And so it's just that we have more columns, instead of one column for each cell, we have, instead of one column for each sample, we have one column for each cell. And of course, because of what I mentioned before, the dropout, this table is going to be even more sparse, right? So even though, whereas here, like you might say like, well, sample 1 maybe doesn't have any of the cell type in it that's expressing gene 1, so we don't see any counts there. Here, like cell 1 here might actually be that cell type. It might be the CD4 positive T cell. Gene 1 might be CD4, but you just, because of dropout, you're not seeing it there, right? Whether technical dropout or biological dropout. And so the sort of processing steps, we saw this last week, the processing steps for bulk RNA-seq, you do the pre-processing in QC, you do the mapping and the counting. So for single cell RNA-seq, it's pretty much the same thing. We're just going to add in a step of assigning a read to an individual cell and then assigning a read to an RNA molecule, right? So we're demultiplexing at the cell level and, essentially binning at the RNA molecule based on the UMI so that you're not double, triple, quadruple counting reads that actually came from the same RNA molecule because of PCR duplication. We went through this last week where we, for the pre-processing, we did FASTQ, multi-QC, Traumatic, and then we ran STAR for mapping and counting. And for single cell, again, this is assuming we're using 10x, it's actually a little bit simpler because 10x has these sort of black boxes. It has this program called Cell Ranger, and then sort of different functions of Cell Ranger. This Cell Ranger make FASTQ is going to actually do the demultiplexing, and sometimes you'll get single cell data in the BCL format. That's the that's the proprietary format for Illumina. Usually for other types of data, you get data as a FASTQ, but for 10x, sometimes you'll get it as a BCL. And so then you can use Cell Ranger, make FASTQ to convert from BCL to a FASTQ and to demultiplex it if necessary, if it didn't come already demultiplexed or converted to FASTQ. And then for like all this stuff here that we saw for bulk sequencing, the QC, the mapping, the counting, and this demultiplexing at the cell and UMI level, that's all going to happen within Cell Ranger count. Like I said, Cell Ranger itself is this black box, and Cell Ranger count is actually like underneath the hood. I don't know what it's using for like the quality control or for the trimming. It's maybe not Traumatic, maybe it's custom, but actually is running STAR under the hood. If you look when you're running Traumatic, you can actually see the STAR jobs running. But you can't, all of this sort of, as you'll see when we go back to the bioinformatics, you don't actually have anywhere near as much control over this process for Cell Ranger count as we did when we were like doing the steps individually, which has pluses and minuses. The big plus is that it makes running the bioinformatics like simpler, and it's not like totally simple, but it is simpler than what we did last week. Questions about that so far?
Hey Josh,
Yep.
I just wanted to note that recently Star Ranger or 10X, I guess, has said that make FASTQ is going to be deprecated, and they recommend BCL to FASTQ, which is Illumina's tool, just as a
Okay.
for people.
Didn't know that. Which make FASTQ actually like runs BCL to FASTQ.
Yeah, it's basically a wrapper. I think they're just
The
maybe
wrapper.
saying it's more
Cutting out the
handy.
middleman.
Yeah,
Yeah. Okay. Thanks. I didn't know that.
sure.
Okay. Uh, any any questions? Okay. Um, so let's get just like a little bit more detailed. Not a lot more detailed, but remember we've now looked at this is the third time I've shown this to you. Right. So here's the structure of the of a fragment for sequencing for like just in general for high throughput sequencing. So biological sequence adapters, and then for a single index barcode, it's going to be here stuck in the middle of the usually stuck in the middle of the reverse adapter, the P7 adapter. So this is what the magic that's kind of happening with 10x is that you kind of already saw this a little bit, but this is actually what the fragment's going to look like. So this part's the same, right? This P5 and read one part is the same. That this is just like in here, this P5 that this is what's going to hybridize to the oligo on the surface of the flow cell, and then this read one is the is the priming site for the sequencing primer. P7, same thing. Um, it's this, and there's even this sample index barcode. You see that here, because sometimes you want to multiplex 10x samples. Actually, often you want to multiplex 10x samples. And then here's the like read two priming site. So all of that's what we've seen before. And then this is the new stuff. And actually, sorry, this is the biological sequence. And you'll see here it's saying like 98 stars. So that's that the length of this is usually about 98 base pairs, the biological insert. Right. So I told you that you're actually usually sequencing a small chunk of the RNA molecule. It's really just a sort of a tag. Then this is kind of the stuff that's 10x specific. So this is that 10x barcode that we saw before, and then the UMI. And then this is this is usually you're aiming not to sequence this. This is depending on which kit you're using, this can be a poly DT, which is used to prime off the polyadenylation on eukaryotic messenger RNA. So usually you're not trying to sequence this. It's it's in the structure here, but the sort of default for sequencing 10x data is that you have a 26 base pair, like 26 cycles, primed off the R1 primer. So you sequence 26 bases, that captures the 10x barcode, that cell level barcode, and the UMI, right? Because the 10x barcode was 16 bases, the UMI was 10. And then you'll usually do, actually I misspoke before. You'll usually do, I think it used to be 75 base pairs. This is saying, again, 98, that you're priming off the R2 priming site. And so you're just sequencing a little bit of the RNA, right? So R1 is in general not sequencing the biological RNA at all. It's just sequencing the barcodes. And then if you did multiplex samples, then you're also going to have a separate set of cycles to sequence the sample index barcode. Does that make sense to everyone? Questions about that? And then like on the back end, the sort of bioinformatic analysis, of course, we sort of already talked about this, That 10x barcode, you're going to use it to group sequence reads to cells. The UMI, you're going to use to group sequence reads to individual RNA molecules within that cell. And then this bit, you're going to map it to the genome, just like we did for bulk RNA-seq, to say like, okay, well, this read came from this gene. And we kind of already saw this before. You're gonna, in general, you're gonna have three files coming off the 10x sequencing. You're gonna have the R1, the forward reads that is going to be the barcode, the cell barcode and the UMI. You're going to have the R2, this set of reads that's the biological RNA itself, and then you'll have an index file that has the sample barcode, and you'll see that in the data that we were using. Okay, so I have a few other kind of slightly more picky details that I think we have time to talk about if there aren't any questions about any of that stuff. Actually, we definitely have time to talk about it. And maybe even have time to talk about it before we take a break. Any questions about all that stuff that I just talked about? Okay. So some details that I glossed over before. So this is the story of Goldilocks and the three droplets. So I kind of touched on this before up here, but I mostly glossed over it about the fact that, you can get, your goal here is to get a droplet with one cell and one bead. You will get some droplets with cells but no beads. You will get some droplets with beads but no cells. You're going to lose some of the cells. They're not going to get incorporated into droplets. All of that is fine. Where we run into problems is so this is a droplet with a bead and no cells. This is another droplet with a bead and no cells. Here are two droplets with one bead and one cell. That's a problem, and this is a problem. A droplet with a bead and multiple cells. Because the problem is that we're making the assumption that when we see, remember, a bead represent all the oligos on a bead have the same 10x barcode. So our sort of basic assumption was that 10x unique 10x barcode is telling us that all the RNA molecules with that 10x barcode on it came from the same cell. If we get two cells in a droplet, then the RNA from both of those cells are going to have the bead E cell barcode on them, but of course there are two cells there, right? It's not a problem for the droplets that have a bead and no cell, and it's not a problem for any droplet that has a cell and no bead, because both of those are just going to be invisible. But this is a problem, and so it's actually a problem that it's a well-known problem, and actually, there's some analysis that's been done that these are called multiplets. Most of what people think of is this problem with two cells in one droplet, but you can have more than two cells in a droplet also. And so you can do some sort of theoretical calculations based on like a little bit of, basic probability, and also like knowing, having some experimental data, and that depending on the number of cells that you collect for a given sample, you have different expected rates of multiplets, right? So if you have 500 cells, you expect about a 0.4% of the cells coming out at the back end, like bioinformatically, about 0.4% of what looks like individual cells will actually be multiplets. So droplets with um, two or more cells in them. Right? When you go all the way up to 10,000 cells that you're collecting from a sample, you expect about over 7% of them, of the putative cells, to actually be multiplets, right? Whether that's a doublet or triplet or whatever. So this is, of course, a problem. So there's some bioinformatic methods. So there are experimental methods to deal with this, which the main experimental method is to just collect fewer cells, but of course that gives you less data. There's there are kind of some tricks that you can use with the feature barcodes to kind of make it a little bit easier to detect multiplets, although it's not 100%. But then the other option is, which essentially you always do, is to do some filtering, use bioinformatics analyses to try and detect the droplets that have or the cells that are really multiple cells. The 10x barcodes that represent multiple cells. And the major way is just looking at sort of the distribution of different genes that are expressed per cell, and the distribution of different unique RNA molecules, so like the unique UMIs, and things that are outliers for those distributions. You basically you assume that those are multiplets. There's steps in the analysis pipeline that allow kind of do some multiplet filtering, and so it's somewhat more sophisticated than what I described, but that's the kind of the basic idea. So other details that I glossed over is that if you're using Cell Ranger for the analysis, so that most common when you have 10x data, you'll be using Cell Ranger. There are some other pipelines for doing this analysis, but if you're using Cell Ranger, then you actually need to, in addition to giving it the sequence data, you need to give it reference genome. We talked a little bit about this last week. And for 10x, you can't just give it like a basic reference genome. It actually has to be like formatted in the, for Cell Ranger, the reference genome has to be formatted in the sort of Cell Ranger format. So 10x has some sort of pre-built reference genomes that you can download from their website, and that's what we use. We've already downloaded it for everyone to use for today. So that's sort of the easy way. But if you have, they don't have reference genomes on their website for every single genome out there. They have like this common things like human, mouse. Maybe they have like, I don't know, zebrafish, rats, things like that. But if you're working on duck-billed platypus, I think that they don't have a duck-billed platypus genome on their website, but there's a tool within Cell Ranger that allows you to take a reference genome and the annotation files and upload, and process them into the format that Cell Ranger wants. So I have switched to my RStudio on DCC On Demand. You will, so we'll take a look at the results of that. Actually, before we take a look, I just want to walk through again what we ran. So this first chunk here is just loading some variables from the config file, like we did last week. This is just basically printing the contents of one of those variables. That's going to be helpful for us in a minute. And then this is just like confirming that we're actually going to be running things with all the CPUs and RAM that we started the container with. And so this is the actual cell ranger command. One of the like weird quirks of cell ranger is that it outputs its results into the directory where you run it. That's kind of like weird and annoying. Most like decent software will let you specify the output directory. Cell ranger does not. So there's a trick to working around that, which is if you want to be able to specify this output directory for cell ranger, you just change into that directory. We didn't really talk about Unix commands, but this cd is a command for changing directory. So you just move into the directory where you want the cell ranger output. This is defined, the cell ranger out dir is a variable that's defined in the config file that we loaded here. So we're just going to change into that directory before we start running cell ranger. Then, this is running, this is like the main event. We're running cell ranger count. Some of these parameters are required and some of them are optional. And some of them I remember are required and some of them I don't remember. So this is this --id and --sample. You can see we're giving it the same information in the library, and that I think that might be used to name some of the files. I don't remember, but basically, oh no, sorry, actually, I misspoke. I think sample is used to tell it. If we look at, let me just look at it really quick. No, it's okay. We can do ls, which is going to list the contents of the directory. So this is going to show us all the files in this directory. So this is where the raw data is. And you can see this is the sample identifier. And then for each sample, if you look carefully, there's actually, oh, I misspoke. So each sample has 4 files. This I1 and I2 are the barcode indices, and then R1 is the forward read and R2 is the reverse read. And so one of the arguments that we're going to give to cell ranger is this -- fastQs. That's the directory where the data is. And so you can see that we have 6 different 6, 1, 2, 3, 4, 5, 6. So 6 different samples represented here. So 24 total FASTQ files. And so this sample here is telling it, okay, well, actually, that's specified here so that we want to run it on this sample. That's the second one here, that this set of files, so that it knows. So cell ranger knows, like, okay, well, which of the files in here to look for. And so it's going to just look for files that the name starts with SR186100021. So that's what the sample is. And then I think the ID is used to like name some of the output files. And we're just going to use the same name for both of them, which is something that we printed up here just to like check it. Okay, so that's ID sample. Transcriptome, that's telling it where to find the reference genome. And that's, again, that's something that we downloaded and put in the sort of shared directory where the not the exact same directory, but basically the next directory over from where the FASTQ files are. And that this variable ref 10x is defined in this config file that we load up here. I just told you what the FASTQ dir is. It's telling cell ranger where to look. This is telling cell ranger how many CPUs to use, because it can actually process these files, like divide up the job and process parts of it simultaneously. So if you remember up here, we checked when we started the container, we asked for 16 CPUs. And so here we're checking that's 16 is embedded in this shell variable cell ranger threads. And so when we print it here, it's 60, that value of that is 16. We're using that same variable, that cell ranger threads, we're passing it to cell ranger here. And then similarly for local mem and local vmem, is we're giving this cell ranger mem value, which is we're printing it up here at 64 gigs of RAM, 64 gigabytes. And then it wants to know like basically which specific library preparation kit was used. That's what this is. And then this last thing, there's actually new with this version of Cell Ranger. If you want it to, it can output a BAM file. You might remember that's like kind of one of the intermediate files that gets output that has the sort of mapping information where each read goes in the genome. For this, I just set it to false because we don't actually care about that. For some applications, you might. And then as that ran, Cell Ranger output all this information, you can kind of get see a little bit like this is listing different versions of different tools that Cell Ranger is running. So you can see stars in there. There's some other packages that it's running. This is a whole bunch of output that we don't really care that much about. And then at the very end, it's going to tell us different things that it output. And so this is the like main output directory. And then within that main output directory, it's going to it used that label that we gave it, or that I guess is the -- ID here to put all the output files in a subdirectory of the kind of main output directory with that same name. And that, it's telling us that these are all the files that it's outputting. That's a little bit easier to see it that way. So we're going to look at this web summary in a minute. These are just like some kind of summary measures of the run. And then these other files are files that you're going to be working with in 2 weeks when you do the biostatistical analysis. This is those count matrices that like basically per cell read counts. And it outputs for you the raw read counts, which is just like pure, like all the counts. And then filtered is that it's gone through this filtering step, including that droplet, sorry, the multiplet filtering. I can't remember if I think there is some other filtering that it does by default, but I don't remember what. And Tyler can pipe in if he remembers off the top of his head. And like that's the major stuff that's in here. And the one thing that I want to do is we can look at this, and actually the way that we can look at is a little bit easier actually if we scroll up here. Let's scoot this over. Scroll up here, if you remember. Here, there's this chunk where we printed out report will be named websummary.html in this directory. So if you find that chunk, it should be right after line 26, and you highlight it and you copy it. So on a Mac, it's going to be Command-C. On a PC, it'll be Control-C. And then you click here on the 3 dots and you paste that in there. So on a Mac, Command-V. On a PC, it should be Control-V. And then say OK. that's going to take us to the output directory. Anyone not get here? Need me to do that again? And I will just, to be clear, each of us put our output in a different directory named for our username or net ID. So what you copy and paste is going to be slightly different than what I did. Okay, so is everyone here? Or anyone not here? Or I go to the next step. Okay, so we're going to look at this web summary file. These are like, well, we could look at the metrics file, but most of these files are not like, or actually, sorry, this is a directory, but most of these files and directories are not like, oh, what's in the directory? Oh yeah, yeah. So it's got the matrix and barcodes, and yeah. So this is information that we need for the downstream analysis. Oops. So I'm going to click on the web summary.html, and then it's going to ask me, do I want to open in what editor or view in a web browser? I want to do view in a web browser. And so this is the basic, this web summary is the like basic output of sort of basic QC summary statistics of Cell Ranger. So this is, a lot of this is like pretty straightforward. This is it's estimating the total number of cells that it observed based on the number of unique cell barcodes. And then this is the estimated number of reads per cell. And then this is the estimated number of genes that were expressed per cell. So about 28,000 reads per cell. And then, the average cell is expressing about 1,900 genes. There's some more detailed information here that we can look at in a second. But I do want to look at this plot because this is actually kind of important. What Cell Ranger does, which is another sort of, this is actually something else that happens in the filtering, in that filtered output. I'd forgotten about this. So this is a plot of number of, let me think. The number of cell barcodes on the, no, is that right? Number of UMI, I think this is the, this is like a barcode, essentially a barcode index, right? So this is barcode 1. This is cell barcode 10,000. And basically it's sorted by the number of UMIs observed for that cell barcode. So this all the way at the top here at rank 1 is a cell barcode that has 31,000 UMIs observed in it, right? And remember, each of those UMIs represents a unique RNA molecule. And so as you go to the right here, of course, because it's sorted by the number of UMIs observed for that cell barcode, the number of UMIs decreases. And there's a point at which it kind of drops off. So this is down to at here, and I don't know if I can get the last one, but it's 618 UMIs. So the Cell Ranger is using this plot to demonstrate like what it's doing to determine the cutoff for cell barcodes. Because see down here, these all in what's shown in gray all represent cell barcodes, but it's cell barcodes that Cell Ranger is saying, okay, well, these cell barcodes, these are actually not, these are probably droplets that didn't get a cell in them, right? And because there's so few UMIs associated with these cell barcodes, it's probably just a droplet with just a bead in it. Now, you will get some UMIs from those droplets and some reads from those droplets because, as I mentioned before, you are going to get some cells that are they're going to break open during the processing. They're going to release their RNA into the into the buffer before the droplets are formed. And so that droplet, even though it doesn't have a cell in it, can have RNA, right? But the assumption and so Cell Ranger does this calculation. Some model, I don't remember the details of how it works, and maybe Tyler's going to talk about it in 2 weeks. But basically, it tries to estimate the point at which on this curve, and you can see it's like kind of a little bit after it starts dropping off precipitously, that it draws a threshold, and basically it's throwing out any cell barcodes that have UMIs below this threshold. And so in the unfiltered, the raw counts, it's got all the cells, but almost always we're just going to work with the filtered cells, right? And so that there's a point here where it's like pretty unambiguous, right? Here you have 4,000 UMIs. Somewhere down here where you're seeing 521 UMIs. It's still calling that a cell, but that means that it's actually only saw 521 unique RNA molecules from that cell, which is there clearly are going to be less than more in a given cell. There are going to be more than 521 RNA molecules. Some of that is just that stochasticity of sampling of what got sequenced, but those are kind of iffy cells, and you can see that there is like this color gradient here. I'm not sure what the blue means, other than it's kind of borderline. So like this is some more summary statistics. Again, this estimated number of cells that we saw up here is from this, basically the estimated number of cells that it observed. The fractions of reads of the total number of reads, the fraction of them that are from what it's considering cells is 96% of the reads are from cells, and then the other 4% of reads are from these what it calls background. These numbers we already saw. Oh wait, mean UMI counts per cell. Oh no, that's not up here. Yeah, so mean reads per cell is right there. Mean UMI counts per cell was 4,700. Mean genes per cell we already talked about. The total, this is like across all the cells, how many different genes did we observe? So it's 22,000 genes. That's about all the genes in the human genome, well, depending on who you count. And then like here's some more summary statistics. That's the total number of reads for this whole sample. This has got some mapping information. You can look at the command line argument. That's what we actually use to run it. And then there's this other tab that it's fun to look at, but it's actually a little bit useless, because it does some of the like sort of visualization that in theory you might want to do, but this is it's just sort of configured. Cell Ranger is configured to do this, kind of the same for everything. And of course, it doesn't know anything about our samples, right? And what we would consider like reasonable. So like it's doing some clustering here, or doing clustering here it's the same projection. We'll talk about that in a couple of weeks. Same projection here. It's colored by the total number of UMIs. Here it's colored by, it's trying to cluster the cell, like I talked about before, kind of group cells into cell types based on their gene expression. Here it's saying for a bunch of different genes, like what the expression is in the cell. You can see like cluster 1 looks like it's probably CD8 positive T cells. It's got a fair amount of expression of CD8A and CD8B. Here's some other information about, well, this is a summary of that figure that we saw before, the median genes per cell. And then like here, it's basically saying like, if we wanted to like kind of exhaustively sequence this sample, we'd want to have like sequenced it more. but that's okay. And that's about it that I have to show you. Are there any questions beyond that, beyond what I told you? About cell ranger, about single cell RNA-seq in general. Like I said, so next week, you're going to be working with Dr. Shi. Um, uh, um, um, she's going to be going through uh, statistical analysis, downstream statistical analysis of bulk RNA-seq. And some of that's going to be relevant for single cell RNA-seq. And then the 2 weeks after that, um, Tyler is going to be walking you through taking the output of Cell Ranger and doing like the downstream analysis using primarily Seurat to do this sort of visualization, some sort of visualization like this, using a somewhat different method. And I think you're also going to be looking at differential expression between, I guess, treatments or clusters, I can't remember, and a bunch of other analysis. And my goal for today is just to get you to this point of having that output from Cell Ranger.